so you're all on camera.
Thanks, Ben. All right, well, it is my pleasure to introduce two students from our lab group today who will be giving updates on some of the work that they've been doing over the last year. The first is going to be Kaci Zarek. Kaci is a junior from Norfolk, Nebraska. She is an environmental studies major. I don't think any other doubles or anything, right, Kaci? Double majors. And an honor student, and has been working in the lab since her very first day at KU. First as an emerging scholar student for 2 years, and now embarking on an independent project, which happened to be very independent because, as we know, last year was an abnormal field season year. And despite all of that, Kaci still managed to do the project that she'll be presenting on today, which she'll continue working up as she moves through her junior year into her senior year. All right, so I'll do an introduction for you too, Kynser, once Kaci goes. So Kaci, are you ready to start? Mm-hmm. Great.
Hi, my name is Kaci Zarek, like Dr. Bergen said, and for my project, we use fluorescence spectroscopy to characterize dissolved organic matter in eastern Kansas streams. But why characterize dissolved organic matter? Well, global changes such as glacier shrinkage, extreme weather events, land use changes, nutrient pollution are important to study because dissolved organic matter is sensitive to global changes. For my project in particular, we looked at the subsection of global changes such as land use changes, because with increased land use changes like increasing agriculture or restoring native grasslands, this can have implications for dissolved organic matter composition, in turn affecting the carbon cycle, because the bioavailability and the breakdown of dissolved organic matter can be impacted by these different land use changes, thus having ramifications for the water quality. For my project in particular, to characterize dissolved organic matter, we use fluorescence spectroscopy to study the source and the composition of dissolved organic matter. This figure to the left is a nice layout of the different indices that can be used to study fluorescence spectroscopy. On the y-axis, we have the excitation wavelength, and on the x-axis, we have the emission wavelength. Together, this creates an excitation emission matrices, or an EAMS. And an EAMS contains lots of information, such as these different indices noted in the figure, like the humidification index, Higgs, the BIX freshness index, or the fluorescence index, FI. For my project, we specifically use the two indices, BIX and FI. FI, for example, is the fluorescence index. So this provides insight into the origin of organic matter. A value of around 1.8 is more microbially derived compared to a value of around 1.2 is more terrestrially derived. BIX, on the other hand, which is the freshness index, accounts for the presence of biological activity and the microbial influence. So a value greater than 1 tend to have more microbial influence. And the really cool part about FI and BIX are that they are both unitless indices, which is something you don't see a lot of. And these indices are quite common in studies. For example, the study by Wilson and Xignopoulos in 2009 used these indices FI and BIX. However, they looked at a continuous cropland or a gradient land use. And what they found is that with higher percent cropland, there is greater FI values and greater BIX values. And another cool thing to note is how they are both positively correlated. As you see, as over the increase of percent continuous cropland for FI, it increases, just the same with BIX. And so really they got at how percent cropland is related to FI and BIX. Although they looked at this continuous cropland and this gradient land use, for my project, we looked at two different watersheds, croplands and grasslands, and we compared the two. These sites have actually already been chosen and were selected previously for in-stream nitrogen cycling and for ecosystem metabolism by one of our graduate students, Kynser. 3 of these sites have greater than 80% cropland watersheds, while the other 3 of these sites are greater than 80% grassland watersheds. And this map on the right shows how all 6 of these sites are found within the same basin known as the Delaware Basin. 3 of the sites are cropland, located up at the top, are noted by that yellow circle, and 3 of our sites are grasslands, more south of those upper stream cropland are noted by the green triangles. And another important thing to note is how we do know some of the water chemistry. So the nitrate and the ammonium, NO3, NH4 are greater in our cropland sites than they are in our grassland sites. And my project is part of the larger Kansas EBSCOR project MAPS, the microbiomes of aquatic plant and soil systems across Kansas, with hopes to add to the understanding of microbial influence within these streams and how land use changes affect these and the origin of organic matter. Therefore, to compare these two watersheds and understand how land use changes might be impacting the composition of dissolved organic matter, we asked, are there different DOM composition signatures in streams draining primarily cropland versus grassland watersheds? For my prediction models, I thought back to the Wilson and Zignopoulos study and how they showed on their figures and with their results how FI and BIX are positively correlated. So I hypothesized that would be the case for our streams. We would see this positive relationship between BIX and FI. I also believe since croplands are more monoculture and less plant diversity, they would have higher values compared to grasslands due to grasslands having more plant diversity surrounding the streams and thus entering the streams. I also want to note how I predict that there will be more microbial influence in the cropland sites as well as more microbially derived organic matter in the cropland sites compared to the grassland sites. To answer this question, we went out to a lab one day and we collected 4 different types of organic matter, sediment and biofilm, both of which are comparable and found across all 6 sites. So that is the one that we collected from all 6 of our sites. Biofilm specifically is found on top of rocks. So it is green, and in this bottom left corner is a picture of me picking up biofilm. And a funny thing, since I don't have much experience with plant IDing, and so Bri was very crucial in this aspect. One thing I could do well was I was really good at picking up the biofilm, so I had to get a picture of myself thanks to Bri at the end of me collecting biofilm because it was the one thing I knew how to do. And on top of that, we collected corn from all of our cropland sites and then a dominant grass type from all our grassland sites. So the top left corner photo, that is a picture of me actually holding a corn husk we found within the stream. That's pretty cool. And also we collected the dominant riparian or the tree leaf type from all 6 of our sites. I do want to note though, that in addition to all this organic matter collection, we actually found a red bug outdoors. So that was fun and not something you see every day at your field site. So when we came back to the lab, we laid out all of our samples of organic matter to dry on tinfoil overnight. And since sediment was kind of muddy and would take longer to dry, we placed them within a drying oven to dry. Then the following day, I divided up the organic matter types by equal weight, and we put them into 3 mason jars for replicates. So there's a total of 72 mason jars. So many mason jars. I probably looked, I don't know, funny when I was at Walmart collecting so many mason jars. Then we added deionized water to make sort of a sun tea. And so there was an incubation time where we collected water samples for FI and BIX from day 0, day 1, and day 7. From there, we analyzed our leachate organic matter water samples for FI and BIX using a Shimazu RF6000. The bottom right photo is a picture of one of the cuvettes we used, and this has a water sample. And as you can tell, there's something funky going on, which I can't really explain, but not in focus is the instrument behind it. In addition to that, we also did analyze these samples for the specific ultraviolet absorbance spectrum. I just want to note this because this happened at the same time as when we ran the FI and BIX, but that will be part of the overarching aspect of my project. A funny story time is that this photo of Dr. Bergen and I shows how we made this project work, especially during a pandemic. So there was one time where Dr. Bergen wanted to teach me a filtering technique that I really had no experience before because I didn't realize how many filtering techniques you could possibly have or do. And so Dr. Bergen came in and she was in the lab teaching me through this glass window while I was in the hallway. And it's quite an experience and memory that I'm going to have forever, just thinking about how like you can learn science so many different ways, but especially during a pandemic, you really learn how to improvise. So result time. Well, when we created our figures for using the FI and BIX results, we focused on sediment and biofilm since they were the most comparable comparable between our 6 sites. And if you're thinking back to the prediction models, these don't quite add up. Like, we thought that cropland would be higher than grassland, and what we are seeing is, yes, there's this positive relationship between FI and BIX, but cropland and grassland are pretty similar, except when we look at this BIX figure on the right and we're looking at grassland, it kind of has a bit more microbial influence compared to the cropland. Then when we look at the left figure focusing on our fluorescence index, or FI, the cropland and grassland are pretty much at the same point. And so on the y-axis, they're only about around 1, which is more terrestrially derived compared to what I thought. I mean, I did hypothesize that grassland you would see more terrestrially derived organic matter, but I was surprised to see how cropland was so similar in regard to its origin of organic matter. So since land use was really not directly affecting these organic matter types or these streams like we thought they would, we took this a step further and we compared all the different organic matter types to each other. So sediment and biofilm are green, our tree leaf types are the shades of gray. And I do want to note that, yes, Eastern cottonwood, like that bar, it's kind of hard to see in the light gray, which is supposed to note black willow is really light. So my apologies, but also we have the grass and the corns, which are those shades of orange. And what we found is that while land use is not directly affecting an organic matter type, it is indirectly affecting these, because these organic matter compositions varied by their biomass type. And it's really cool to think about how when we look at this left figure and we look at FI, the grasses and the corn seem to have higher FI values. And it's not too close to the 1.8, that microbially derived value. It's kind of in between that terrestrially to microbially derived value. So there's a lot more to think about when looking at this figure, versus the Eastern cottonwood definitely has a lower FI value and more terrestrially derived organic matter. Then on the right figure, the BIX, which helps think about the microbial influence, black walnut stands out significantly, one of our tree leaf types compared to the others. And it kind of shows how there's more microbial influence compared to a black willow, which is also a tree leaf type, or the grasses and the corn. So yes, our predictions did not quite match up like we thought, with cropland having higher values than grassland, but it allows us to think even further about these questions and think, yes, land use might not be directly affecting these, but it has an indirect effect. And what more can we think about when looking at these organic matters? So of course, this entails future work of comparing our leachate organic matter to our stream organic matter, because we collected stream water samples at the same time as these different organic matters were collected. We will also analyze our leachate stream dissolved organic carbon samples, because this will allow us to understand the quality of the organic matter within these streams. We'll use DOC concentrations, then calculate specific ultraviolet absorbance values. There's a lot of specificities to that, so I won't go into too much detail, but it'll really get at that quality of the organic matter. And finally, using our FI values, we shall be able to calculate their excitation emission matrices, or EEMs, and thus give us better understanding of the composition of the organic matter. Overall, thank you to everybody who came today. And I just want to give specific shout outs to my research mentor, Dr. Amy Bergen, for helping me understand so many aspects of my project and making science really fun despite a pandemic, because there's so much you can learn. And also the Bergen Lucky lab mates for helping with sampling and filter. A huge shout out to Bri, who helped me collect these organic matter types and taught me so much about plant IDing that I never knew, really. And Dr. Lucky for helping me create figures of R, because R is hard nonetheless, but learning it remotely feels really hard, but it was still fun to learn. And yeah. And finally, a colleague from Ohio State University, Rachel Gabber, who met with Dr. Bergen and I via Zoom over the summer, and she taught us the beauty of fluorescence spectroscopy, which I got to share with you today. And finally, the funding sources, specifically the KU Field Station Award. So thank you.
Thanks, Kaci. That was really well done. All right, next up we have Kynser Wahwahsuck. Kynser has been around KBS for a while, so it's probably a familiar face. She's a second-year master's student in the Ecology and Evolutionary Biology department. Before she joined as a master's student in our lab, she was in the prep program for a year, which is an NIH-funded program for students in between undergrad and graduate school. She did her undergrad at Haskell Indian Nations University and graduated in 2018. And while she was at Haskell, she was still connected to KU because she is also a Bridge program alum, and I think a HERS program alum, and an RU, I think, at KBS as well, alum. So she's garnered a lot of research experience that prepared her for her master's work, which is what she's going to be sharing with us today. So welcome to Kynser, and you can go ahead and get started whenever you're ready.
Thank you for that introduction. Great. Thank you guys for coming to listen to us today. We both have the same study site like Kaci had said, so you'll see a lot of the same things there, so I hope it sticks. So yeah, like Amy said, my name is Kynser Wahwahsuck, and I'm a member of the Kickapoo tribe in Kansas, and so looking at stream health within this watershed is important to me because it runs through my reservation. So I'll be talking about stream metabolism in Kansas streams draining cropland versus grassland watersheds. So why do we care about stream health, and why should you? The Midwest is an agricultural belt, and farmers put nutrient-loaded fertilizers on their fields and also grazing of cows. The cows also poop out nutrients, and it all has to go somewhere. So it flows down the Mississippi River and into the Gulf of Mexico and creates these dead zones where there's low oxygen, and that's terrible for fish and creates bad quality for recreational use and drinking water quality. And this is happening at a big scale like this, but we're going to take a step back and look at it from a smaller scale. The Delaware Basin in northeast Kansas. This watershed is a mix of agriculture fields and prairie fields, and it all drains into Prairie Lake down here. And Prairie Lake is known for its recreational use and also for its unwanted guests of harmful algal blooms. And this is a study site for another graduate student in our lab, Janae. Sorry if I said your name wrong, Janae. Okay, so I'm focused on the headwaters before it reaches the lake. I have 3 cropland streams and 3 grassland streams. And when I say we. I'm sorry. I feel bad. I don't want to leave anybody out. There's a lot of people that work on this project. And we have 3 cropland streams in the blue. And they have their respective watershed size that drains into each stream, and the percentage of cropland and grassland within those respective watersheds as well. And we have our big question is, how does land use affect in-stream denitrification, nitrogen concentrations, and ecosystem metabolism? So what we did to tackle this question is we did a series of 24-hour experiments among each stream. We did this to measure diel patterns of nitrogen concentrations. We took samples every hour on the hour, and this was to estimate different forms of nitrogen to quantify nitrogen cycling processes, such as denitrification. So during this presentation, whenever you see this colorful timeline, that is referring to the 24-hour experiments. And you can see many pictures of us in working hard. And so here was our setup during each experiment. We had our sample station where Norma is. She helped me during most of these 24 hours. She was a student at Haskell that got to come work with us during the summer. And that was our sample station where we took water chemistry samples, gas exchange samples, as well as we had some sensors close by measuring nitrate, and we had the YSI. Further upstream, we also have a sensor array set up that is measuring dissolved oxygen, conductivity, stream water level, water temperature, and this was measuring every 30 minutes during our 24-hour experiment. And we have one of these sensor arrays set up in each stream within the Delaware Basin as well. So before we did our data analysis, we had some predictions. One being our nitrate concentrations. Our grassland streams, we expect to produce shallow concentrations due to the overall lower supply of nitrogen coming into those grassland streams. And we expected our cropland streams to have higher nitrate levels of concentration with this diel pattern I had mentioned before. And what we mean by that is that our the concentrations would go down while the microbes eat up the nutrients during the day, and it would go back up through the nighttime. And also, we just expect to see overall higher concentrations of nitrate compared to our grassland. As well as with our denitrification rates, we expect our cropland will be relatively higher than our grassland land use. So next, I'll show you what we did find. Our chloride concentrations stayed relatively stable throughout each 24-hour experiment. This supports the rest of our data in showing that there wasn't a big disturbance in our stream flow, such as a rain event. And next we have our nitrate concentrations in milligrams per liter, and on the bottom axis, if you can see it, there is a timeline again, colorful timeline. And so our cropland concentrations ranged from 10 milligrams per liter to 30 milligrams per liter, which was relatively higher than our grassland streams that ranged, or well, they were less than 10 milligrams per liter. So this is likely due to the amount of nutrients coming into those cropland streams from agriculture. Next, we have our GPP, our gross primary production and ecosystem respiration. We had higher rates of GPP in our cropland versus our grassland, and as well as in the cropland, this might be tricky to explain, but our ecosystem respiration is in its absolute value, so it's flipped. But looking at it the other way, there was more ER happening in our cropland streams than our grassland streams. And we can compare this to other previous studies, such as Michelle Kelly's master's thesis. The colored dots on here represent her data points in the Kansas River, which is a much bigger system than the headwater streams that we're working in. On the x-axis, you can see it's measured by discharge, and then we have GPP and ER on the y-axis. And so again, the colorful dots are her data points, but if we, let me take a step back. The gray data points are a whole bunch of previous studies and where they fall along according to their discharge size. And so if I put my data points in here, they fall right here. And so next we'll look at denitrification rates. Our denitrification rates were higher within our cropland streams and not so much action in the grassland streams. And again, I'm going to compare this to other studies. This is AJ Reisinger's 2015 paper, and let me explain his graph a little. So this one on the left is based on methods, and my data will fall on the open channel method along with 16 other studies and denitrification. And then this is the habitat scale, and mine again falls under stream and river. And then if we put my data point in with this and 137 other studies, my data falls along with. So in conclusion, our chloride concentrations were relatively stable, which supports all of our 24-hour experiments that there were no big disturbances. Our higher nitrate concentrations were likely due to fertilizer and grazing runoff into those cropland streams. And our gross primary production and ecosystem respiration show a difference between land use type that may be due to light or nutrient availability. We haven't looked that deep into this yet. And then we have our denitrification that differed between land use, and denitrification is typically limited by availability of nitrate. And so that's why we believe that we see more denitrification happening in our cropland streams as there is more available nitrate. And thank you so much to everybody who's contributed to this project, whether it be doing the 24-hour experiments, going to check out the sensors and doing data processing, helping me program with R, like Kaci said, because yeah, it's a tough one, and helping me go through my talks like this. Thank you to everybody.